We didn't cover all of the slides of chapter 3, but I mean, you have those, you can look over them. We're going to cover DNA in a great amount of detail in a couple of weeks. So we're going to transition over into chapter 4, and this is an overview of the cell. And if you've already looked at this slideshow, you realize that there are like 50, I think it's 54 slides, 55. It's a lot of slides. However, we're going to move through it pretty quickly because it's kind of the overview. Again, like this 40,000 foot view, and then we're going to go in and we're going to dig deeper. Okay, so first set of questions. Hopefully we'll get through these 4 questions today, and then maybe some in the next set. But first question: what seems to be true of all cells? Whether you take a cell from a human or from a guinea pig or something really awesome like a fruit fly, right? Or you take like a cell from just a single-celled organism. What is true of all cells, no matter where they come from? Nothing on the fruit fly? That was hilarious. Thank you. How do we view microscopic objects? I mean, that's such a wonderful question because the answer is right in the question. We use a microscope. What can cells do to ensure proper transport and function? We're going to have a whole chapter on this. So this one, you're going to be a little dissatisfied with our answer because you're going to be hungry for more. We'll give you plenty more, right? You're going to get less than like the little finger things you get in the hot appetizer hour at somebody's wedding. The reception was at 1 o'clock. So they're like, oh, we don't have to give them lunch or dinner. We'll give them little hot appetizers. How are prokaryotic cells unique? All right. Okay, so what seems to be true of all cells? So a little bit about the history. I don't often do this because I think science is more important than scientists, but every once in a while I will show you very specific people because they really transformed the way we operate as scientists. Okay, and so Robert Hooke was one of these individuals, and Robert Hooke was the first person to actually take one of the most wonderful technological materials of the day, being the ability to see things really small, and decided to look at something big, right? To look at something big with a tool in which you can see things really, really small. And what he did was he looked at cork, which is a type of plant, and he saw these, what he thought looked like jail cells making up this cork. And so he postulated that living things are made up of cells. It's so wonderful. Van Leeuwenhoek continued to improve on this design and then used it to start to demonstrate that what people had just kind of ascribed to these random acts of magic, he postulated that no, there are probably living organisms doing these kinds of things. Like, you know, if you put a piece of raw meat out and it didn't get colonized by flies, it would still eventually disappear. And, you know, it's not some random act of magic. Leeuwenhoek's like, no, there are probably these really small organisms that are eating that material just in the same way we eat material. And again, really transforming how we think about the natural world. And then Robert Brown describing the nucleus, so thinking not just like, wow, these are these cells, but thinking, I bet there's something inside of these really tiny structures, and they're not just this bag of nothingness. And so from this work, you get what's called the cell theory. And this is the first time we have this opportunity to talk about this, the difference between the word theory and law. Okay? True or false? A theory can become a law, given enough evidence. Raise your hand if you think true. Oh, this is so awesome. Because you've all been taught wrong. A theory and a law are completely different. A theory is describing why, a law is describing what. Okay? A theory cannot become a law no matter how much evidence there is to support of it. It's always still a theory because it's describing why something happens. Okay? And so here, the cell theory, this is misnamed. This has actually been misnamed by somebody that was in your boat because this is actually describing what? It should be called the cell law. And in some texts, you actually see it described as the cell law, but in our text, it calls it the cell theory. Because this is describing what? All organisms are composed of cells. Need like this? Of cell or cells. Because some living organisms, they're just one, right? You know what? Yeah. The cell is the smallest unit of life. If you take anything from within that cell and remove it, it is not a living organism. Okay? Because the cell is the smallest unit in which you have all of the characteristics of life. Okay? Anything within a cell is going to be missing some of the characteristics of life. And then finally, cells come only from pre-existing cells. Cells come only from pre-existing cells. All right, so these are the three components of the cell law. Okay, of the cell law. Any questions about this? So we're going to finish up this question, and then I want to do a little bit of an exercise that I hope you'll find fun. At least I hope you'll find enlightening. All right. So what seems to be true? In addition to all living things being made up of cells, all cells carry some form of nucleic acid. If they do not, then they cannot be considered a cell. And so this gives you the idea of like, okay, the cell is the smallest unit of life, and cells have nucleic acid, so it gives you kind of the idea that all living organisms contain nucleic acids. That would be the exact feeling you should have, the thing you should associate. Also, cells metabolize, they acclimate, meaning they respond to stimuli. If you heat a cell up, it's going to respond accordingly. If you put a cell into a different type of environment, it's going to respond accordingly. And cells reproduce. So again, this is giving the idea that living things need to be able to metabolize. That is, they carry out chemical reactions. Living things need to be able to acclimate, respond to stimuli. And living things need to be able to reproduce. If it can't do these three things, it is not a living organism. All right? All right. Now, additionally, in multicellular organisms, cells also display division of labor. What does that mean? Yeah, we're the same, basically the two different cells have to be capable of doing different jobs, right? And not because they're different genetically, but because they have to carry out different jobs, okay? Because in a multicellular organism, all of the cells in there, not all of the cells, but all of the cells that belong to that organism descended from a single cell, the zygote, okay? They all descended from that cell, meaning they're all genetically identical, and yet some of them are doing different jobs than others and actually look different than others. And we'll talk in a few weeks about how that actually happens. How do you get two cells that are genetically identical, one to be a liver cell and one to be an epithelial cell? How does that work? Or even more complicated, how you can get one to be a liver cell and one to be a neuron. Okay, how does that work? Okay. So, any questions about this one, of what seems to be true of all cells, regardless of where they come from? Okay. So what I want you to do is this. We're going to take our lecture break. It's a little bit early. I'll try to do a second lecture break. And I want you to take about a minute, and I want you to go over the three parts of the cell law, and I want you to tell me which one has to be violated. Which part of that cell law has to be violated. Okay? So take about a minute, go through those three components, talk with those around you, and I want you to tell me which of those three there has to at least been once in which that was not true. All right? 1 minute, starting now. And then give me a why as well.
So it's like this composite process.
Right. If you're saying cells coming from cells, I would.
Because it seems like the other two are opposite ways. Really? Their definition is You definitely like spent like the entire unit talking about like 5 hours in molecules. I don't think that
All right, let's go point by point. All living organisms are composed of cells, or all living things are composed of cells. Did there have to be a point in time in which that was violated? No. Even the idea is if you're going to get life from non-life, you're going to get life without a creator. There's still the idea that you really don't have a living organism until you have a cell. Okay? The nice thing is, and we'll talk about this next week, that making just a very simple cell is actually not that difficult. It's making a functional cell that becomes difficult. Anyways, it's another story for later. What about the second one? That the cell is the smallest unit of life. No, that one's very similar to the first one. We don't need to violate that. But what about that third one? All cells come from pre-existing cells. Eventually, you've got to have the first cell, right? Eventually, you've got to start the process. And so it's a nice thing to think about, and you're just like, oh yeah, the cell law. Like, we can explain how we get new organisms from other organisms. They just came from other cells, right? But eventually, you have to get to the beginning. At some point, regardless of your view of origins, you have to violate the cell law. Initially, you have to have the original cells either created, or you had to have just the first cell that formed from inorganic materials, or a combination of organic and inorganic materials. All right? At some point, you have to violate that law, that portion of the law. And that's an important thing to keep in mind as we talk about the different philosophical commitments that apply to cell biology. Okay? Because I'll tell you this. The world was ready for Charles Darwin to publish on the origin of species and to provide an explanation of how you could get the diversity of life without a God. The world was ready for it. There were already atheists before Charles Darwin published his On the Origin of Species. They didn't have an explanation for how that could work. Okay, but they were already ready for it. But one of the reasons why it was so successful is because nobody knew how complex cells really were. If we would have waited until today, or even 50 years ago, to publish that, it would be a really hard sell. Which is why when you... hard sell, I didn't even mean to do that. Wow, that was awesome. It's the reason why there's an organization that exists to basically ensure that evolution is taught to every human being on Earth. Okay, there's an organization that invests lots and lots of money to ensure that that happens. They recommend that you always teach somebody evolution before you teach them molecular biology. You have to teach them evolution first. And there's a real reason for that, because you need to be committed to that before you learn the complexities of molecular biology. Anyways, that's a story we'll continue throughout this semester. So here are the three major types of cells, the three domains of life. These two are prokaryotic, which we'll talk about on our fourth question. This cell right here is a eukaryotic cell. And so you should notice something right away looking at this, that this has, that you don't see here. You see this? The nucleus. Okay? And so it's just something for later. So here are several more eukaryotic cells. If you look in these, this cell is very strange, but you can see the nucleus in there. This is highlighting the cytoskeleton. That's why it looks very strange. Here are animal cells. You see the nucleus, plant cells, fungal cells. And so in eukaryotic cells, there is something very significant inside of those that you do not find in prokaryotic cells. And it is the nucleus, which is where those names come from, by the way. Prokaryos means first nucleus, which is kind of fun because they don't have one. And then eukaryotic means true nucleus. A little bit for later. All right. So how can we see microscopic objects? This is the best question to be asked. It's like, what color is an orange? But where it gets into is, what type of microscopy? Because there are different types of microscopy. So let's just run through this really quickly. So in order to use light microscopy, what you've done before, where you take a microscope, you put something just that is just so intriguing underneath it, and then you look at it, like you're awakened to how tiny life can be. These light microscopes, they need basically visible light in order to be able to display an image, to create the contrast necessary for you to see it. The smallest wavelength of visible light is 400 nanometers. Any light with a wavelength smaller than that is not visible light. Meaning the smallest objects that we can see using light microscopy are things that are 400 nanometers. Like, that's really small, but there are living structures that are smaller than that. And you'll see why we talk about living structures in a minute. On top of that, light microscopy is limited by the fact that you have to stain something. So it's very difficult to use light microscopy on something that's still alive. I mean, we do it, but it's hard to see it, and it kind of sails through really quickly. And unless it's photosynthetic and it's green because of all this green pigment it has in it, it's basically like see-through, right? And so you see something see-through move through the field super quick, and you're like, cool, I saw something. Don't know what it was, but I saw something, and it looked alive. So light microscopy has its limitations. And then we get electron microscopy. So electron microscopy, it doesn't using light, or it doesn't create an image using light. What does it create an image using? Electrons. How do you know? Because it's called electron microscopy. It's so wonderful, right? Another example of where the name makes sense. The wavelength of an electron is way smaller than the wavelength of the smallest photon of visible light. Okay? And so the resolution of electron microscopy is much, much better than light microscopy. You can see structures much smaller than you can with light microscopy. And so when we started using electron microscopy, really started using a lot in the 1980s, it was amazing to see how much detail there is on the outside of a cell. Because it's just kind of like this idea of like, oh, it's just a membrane. But then you look at it and you're like, wow, there are a lot of really complex structures on this membrane. So electron microscopy is limited in the fact that it usually is really just effective for viewing the surface of an object. You do have a different type of electron microscopy called transmission electron microscopy, but it's a little bit more complicated. And this is very expensive. If you want to look at the outside of a structure, you have to coat it with some kind of a metal. So you've got to kill whatever it is that you're looking at, and then you've got to coat the outer surface with a metal, and then you can basically beam it with electrons, and as the electrons are deflected, a computer screen picks them up and builds an image for you. I've got some really good electron microscopic images of the telson of scorpions. So the telson's the stinger of scorpions. And so I've done a lot of work with scorpions. I've got some pretty neat scanning electron microscope pictures that I'll show you when it's time in lab. I know. So here's light microscopy. Here's the standard light microscopy that you're familiar with that we use in lab. It's called bright field because you're basically shining a whole bunch of light, and where the light gets absorbed, you see that as an image, okay? But if the light has a smaller wavelength, or if the light, only light with a wavelength smaller than visible light gets absorbed, you don't see it because you don't see that wavelength of light anyways, okay? And then here's dark field microscopy, which is similar to bright field microscopy, but you're looking rather than at the places where light gets absorbed, you're looking at the places where light is able to penetrate and move through it, okay? It's a little bit different style of microscopy. Two other types of light microscopy, phase contrast and Nemarsky. They use different ways of focusing the light and different ways of bending the light to create an image. So they're we can use those to pick up structures that we wouldn't normally see because they're not really absorbing light, but they are deflecting light. Okay? And so these rely on that. They basically will take the way light is being deflected and we'll use that to build an image. Okay? Anyways, this is just, if you're like, what do we need to know about all these? Should we anticipate like a matching question? No, I'm just showing you the different, I want you to love microscopy. You're like, if you want us to love it, you should not talk about it. Just say it's great and then say nothing else, right? I get it. So here's fluorescence and confocal microscopy. Fluorescence, you basically, you inject something with a fluorescent dye. You don't give it really any other light, and then you just look for where you're getting that fluorescence showing up, that light emission. And then confocal, you use lasers to beam this object. And we have one of these microscopes. It's pretty awesome. If you want to look at something on it, you can talk to Dr. Francis, and he can set you up. All right. One last thing, electron microscopy. So here is scanning electron microscopy. Notice we're only seeing the surface of these cells. And here's transmission, where we're actually shooting electrons through the object. As you can probably imagine, it's difficult to keep this thing alive when you're shooting electrons through it. Okay. So unfortunately, both are forms of microscopy, electron and light microscopy. You can usually only really look at things that are already dead and fixed in terms of getting really good resolution and a good idea of what's going on inside. So then it forces you to ask, which question? If you know that you can only look at these things after they're dead, what question might you ask?
How do they work?
Right, how do these structures work? Because things don't work after they're dead, right? When a cell dies, it reaches equilibrium. All chemical reactions stop. Okay? At least within that cell. And so structures aren't really working anymore. So then you have to kind of infer how they work based on where they're located. Okay? Or you can do some other things which we'll talk about at a later date. What else might you ask? How do they move? Do the structures change at all after they die and they're no longer being used? Because think about this. If you've got a structure packed full of water and the cell's working really, really hard to maintain water balance and to keep that structure packed full of water, what happens when that cell dies and it can no longer work really, really hard at keeping that structure packed full of water? It shrivels up, right? And it deflates. And you're like, now you might have an idea that the cell looks like this, and it only looks like this when it's dead. You're like, well, so what does it look like when it's alive? It's a wonderful question. But we're getting better at this. We're getting better at studying living, really, really, really small things. Okay? All right. Next question, and then we'll do another lecture break. What can cells do to maintain their barrier and to maintain really good quality transport within and outside of the cell? First thing you need to know, and this is something you cannot ever forget with regards to cells. You really shouldn't forget it with regards to anything, but cell size is limited by the amount of surface area it has relative to its volume. Okay? And I have a figure to show you what this actually means. But the less surface area a cell has relative to its volume, the harder it is to move materials into and out of the cell. And the harder it is to move energy into and out of the cell. Have you ever noticed that a baby always feels super warm? Have you ever held a baby and you're like, man, this thing feels like it's on fire? And then you take its temperature and you're like, no, it's normal body temperature. But it feels so hot. Have you had that experience? Okay, babies have a crazy high metabolic rate because they are growing and developing like crazy. And that's where we generate our heat, is from our metabolism and not by typically absorbing it from our environment. And babies have massive heads compared to the size of their body, right? At birth, a baby's head is like a quarter of its body weight. Okay. for an adult, that would be crazy, right? The average adult head weighs, I think, 8 pounds. The average adult weighs, I don't know, something probably around 140 pounds. Okay, because you got to do male and female and different sizes, right? And so you're like, okay, 8 pounds divided by, let's call it 160 so the math is easier. 8 pounds divided by 160, I believe that's 2%. Right? Because, no, it's 5%. Because 80 divided by 16 is 5, but it's 8 divided by 16, so that would be, yeah, it's 5%. Because 16 would be 10% of 160, right? So 8 is 5. I did that to make the math easier, and then I messed it up initially. So it's about 5% of the body weight. But a baby, it's like 25%. That would be a giant head for an adult. But the baby needs that because it needs to be able to get rid of all that excess heat it's generating by its crazy fast metabolic rate. Okay? That make sense? Okay, cool. All right. Cells are surrounded by a hydrophobic barrier. Okay, and we'll talk a lot more about this when we talk about cell membranes, but we've already talked about a specific type of lipid that is both polar and nonpolar. And what is it? A phospholipid. And so you've got these phospholipids that they will line up. Again, we're going to talk about this in a lot more detail. It's just a little flavor, right? It's like a hot appetizer instead of giving you a sandwich. So just make me a sandwich. I don't need this little like Philly cheesesteak slider when it's only got one side on it, right? It's like a cracker with some meat on top. Just make me a sandwich. It's no big deal. All right, anyways, sorry. So the phospholipids line up to where those hydrophobic regions can be side by side, and you create this hydrophobic barrier. So it prevents water from moving super efficiently across that membrane. So water-soluble, also known as polar substances, these aren't going to move across the membrane very well because you've got this massive non-polar region. So they have to be transported through specific channels, and the cell has to control that. So it gives the cell a very high level of control over what gets in and what gets out. Because here's the thing. So just give me something that's polar. What's a polar molecule besides water? Or it could be just, what's that? Oxygen as a gas, no, is not polar. How about like some kind of a sugar, right? We talked about this, our biomolecules. The only biomolecule type that's nonpolar are lipids, right? So sugars are polar, proteins are polar, nucleic acids are polar. Salts are beyond polar. Salts are ionic. Okay, so those have the same problem as polar molecules do. So yeah, salts, sugars. Let's say you're a cell and you don't want a particular sugar type getting inside the cell. You're like, why wouldn't I want sugar inside of the cell? Maybe you don't have the machinery to metabolize it. Okay? And so you don't want it getting in. The beauty of it is it can't get in unless you have a channel that it can move through. So if you're that cell and you don't want that sugar inside your cell, what do you avoid doing? Oh, either opening the channel or you could just avoid making the channel altogether. Right? If you don't make the channel, that channel can't be in your membrane, that sugar can't get in. It's beautiful. Oh, by the way, I looked it up afterwards. So for those of you on Monday, I was like, wow, this is really weird. The milk should fail the reducing sugar, but I looked it up and lactose is a reducing sugar. So, yeah, that just changed everything. I know. So that's, you know, somebody was like, hey, could we write for what could I have done better, have come up with our own hypotheses? I think that was actually my idea. That actually totally works for this one, because then you probably would have looked up whether lactose was a reducing sugar before you just assumed it wasn't, like I did. Sorry. Okay. Cytosol is the liquid inside of the cell. It's water-based, water as the solvent. And it has a lot of materials dissolved inside of it. A lot of materials dissolved inside of it. Some of those are stored up for later use. Some of those are busy doing some very specific task. All right. And then lastly, oh no, there are two more. Sorry. Then we have the cytoskeleton. We saw this before. Again, this root cyto means what? Cell. And then the cell skeleton maintains the shape of the cell. and functions to move materials inside the cell. Really, they function like roadways for things to move on, but we'll see this in a couple of weeks. And then the term cytoplasm, you've heard that term before, right? Cytoplasm is the cytosol along with the cytoskeleton. So it's the fluid of the cell as well as the skeleton of the cell combined together to form the cytoplasm. Okay. All right. Any questions about this? What can cells do to maintain proper movement of materials? Do you have a question on something else, Levi?
No, just the cytoskeleton.
Uh-huh. Those are made up of proteins. It's one of the functions of our protein biomolecules to make up the cytoskeleton.
So the cytoplasm is just like the
The cytoplasm is the weird material that's made up of a liquid and solid protein.
And then the cytosol is the liquid.
Yes, the cytosol is the liquid component. Yep, and then the cytoskeleton's the solid component within it. And so it actually, the cytoplasm can vary from being liquid to solid. So it can, if you've got very little cytoskeleton in a particular area, it's water with some protein dissolved in it. But if you build up a lot of cytoskeleton in that area, it actually becomes solid with water sprinkled throughout it, like a jello. A gelatin, which is a solid with liquid interspersed to make it, move around a little bit, super flexible. Yeah. Anyways, we'll see that more a little bit later. And so here I want to show you the surface area to volume ratio issue, and then we're going to do a second lecture break. Okay. So here we have a cell. I know this is a cube and cells are spheres, but imagine that you had a cubic cell. Okay? Cubic cells would actually be a wonderful, they're a wonderful design because you can pack them close together, plus they maximize the surface area relative to the volume. Then why aren't cells a cube? And it's because it would be very energetically costly to maintain a cubic shape. And plus some cells are cubes. Anyway, sorry. So we've got a cell, and it's a cube with an edge length of x. Okay? So if you were going to multiply surface area, you would find the surface area of this square here, and then you'd multiply it by 6, right? So the surface area of this square, if the edge length is x, the surface area would be x times x, which is x squared, right? And then we have 6 of them, so surface area would be 6x squared, right? And then how would we find the volume? We do edge times edge times edge, so we would do x times x, which is x squared, times x, which is x cubed. And so there is our volume. And then so the ratio of our coefficients are 6:1 here. Okay? Now let's double our edge length. Okay, so we've taken the cell and we've doubled the edge length. So if it were a sphere, we would double the diameter. Okay? Now, if we're going to find the surface area, it would be 2x times 2x, which is what? 4x squared, right? And then times 6 would be 24x squared. Yes? Okay, what about the volume? We would do 2x times 2x times 2x, which would be 8x cubed. And now when we do a ratio of the coefficients, 24 to 8 is actually 3:1. So now our surface area to volume ratio has been cut in half when we doubled the edge size. And why is that? Why is that ratio shrinking? What's growing faster, the surface area or the volume? The volume, right? Our denominator. And the faster that denominator grows, the smaller this gets. Look, by the time that you have quadrupled your edge length, your ratio is now 1.5:1. Okay? And why does this limit cell size? Why does this limit cell size? Yeah, Micah.
Well, because you want as many nutrients to be able to get inside to the volume.
Yeah, the bigger the volume of your cell is, the more demand you have for materials to come in, and for the bigger demand you have for being able to get materials out, right? Think about this. What's our good analogy for a cell? What analogy do we love? The factory, right? So we've got a tiny, mom and pop factory in somebody's garage, right? It's like, yeah, they might be doing some cool stuff, but they're not bringing in massive amounts of materials and shipping out massive amounts of materials, right? But now you've got something like a massive 700,000 square foot factory run by sweet robots, right? And so now you've got a much greater demand for materials and a much greater demand to get what you're making out. And so now it's not quite as simple as like, oh, it's cool. We'll just have it shipped to a mailbox in front. They'll just open up that mailbox, take that material across the door, right? It's totally cool. It's like, no, it doesn't work that way at all. Now it's super complicated, super complicated. And then at the same time, you have less surface area relative to what's going on inside of the cell, okay? But this is also true for entire organisms. This is why as a mammal gets bigger, its metabolic rate tends to get smaller, okay? Because the bigger a mammal is, the less surface area it has relative to its volume, right? And so if it was still generating just as much heat, it would have a very hard time getting rid of that excess heat. Would you agree? And so I read this one time, and it's kind of fun. It doesn't directly relate to cell biology, but I want to share it with you. So do you all know what the smallest mammal species is? It's not tardigrades are the smallest animals, but the smallest mammal. It's a shrew. And there are shrews that as adults weigh 2 grams, which there are 450 grams in a pound. This is a small mammal, right? This is a small mammal. That's an adult, okay? If you were to slow its metabolic rate down to what our metabolic rate is, right? Ours has to be smaller because we have less surface area to get rid of all this heat we're generating. If you slowed that shrew's metabolic rate down to ours, its fur would need to be 6 feet thick to keep it warm. I don't even know how you could take a 2-gram shrew that's like this big and you've got fur 6 feet thick. It would look awesome, but I don't know how you would do it, okay? And then if you took an elephant and you sped its metabolic rate up to ours, it would die of heat exhaustion in about 30 seconds, okay? So just kind of putting this into perspective. So this is what I want you to do during this lecture break. So we talked about how can cells get materials in and get out. And so what I want you to do is this. I want you to take about a minute working with those around you, and I want you to just, without using any notes, just what you can come up with, I want you to draw what that membrane is going to look like, okay? I don't want you to use any notes, just draw what that membrane is going to look like, because we've already talked about that if you're going to move something across it that's polar, what does it have to go through? Some kind of a channel or a tunnel, because it can't move through that membrane. Okay? So I want you to take about a minute, don't look at your notes, and I want you to draw what this membrane is going to look like. Okay? 1 minute, starting now.
Let's go, Art Storage.
That's how it's gonna look like. Can't use that.
It's gotta be like a because
The next slide. The next slide.
secondary
All right. So, this is something that you need to save, store away somewhere, and I want you to make this a regular practice of yours to draw features of cells using no resources. Okay, just giving you kind of maybe, I don't know, a little foreshadowing of something you might be asked to do on midterms and final for this class. Okay, and so right now, cell membrane. I'm going to show you a ridiculously simple version of a cell membrane. So here's a cell membrane. We've got 2 layers of phospholipids, right? And they are oriented such that these non-polar hydrophobic tails can face each other, but it created this massive non-polar region, right? And what can't move through there? Anything polar, and especially not anything like a salt, which is what, Levi? It's even beyond polar. It's ionic. It's ionic. It's ionic. Water is super mega polar. It's ionic. It's beyond that. Okay, and it can't move through here. So then we've got these channels that it can actually move through. And if you were to look inside at what types of materials are proteins made of? Amino acids. And if you were to look at these amino acids inside, do you think the amino acids would be polar or non-polar? On the inside of this channel. Polar, right? And that's what allows polar materials to move through it. What about the amino acids that are on the outside of this tunnel? Non-polar. And you're like, how is one amino acid polar and another one non-polar? What changes from one amino acid to the next? The R group, right? Which it could be various functional groups. So what kinds of R groups would make a non-polar amino acid? Something that's non-polar, right? So you're probably going to lack any functional group on that R group. And what is it going to be? Just a big old hydrocarbon. Bunch of carbons and hydrogens, and this massive non-polar structure. So when those amino acids are exposed to here, they love it. The little non-polar R group from those amino acids are hanging out with these non-polar tails, having a nice little water-fearing party. That's on the outside of the channel. On the inside of the channel, you've got a bunch of polar R groups, like with fancy functional groups, that are hanging out with the water and other polar things in here, having a water-loving party. And right next door, water-fearing party. Right here, water-loving party. Happening on the same protein. And this is where we get into that a protein can only function properly when it has which level of organization? Tertiary, which is what? The proper three-dimensional shape. It's got to be folded such that the non-polar amino acids stick out, the polar amino acids stick in. Does that work? I'm going to roll with it. Stick in and stick out. Okay? Is that cool? It's awesome. But why is this overly simplified? We've only got one type of transport protein. Gosh, we need to... What you find is that most of these transport proteins are specific for a very specific material. So there's some that are specific for glucose and some that are specific for some other polar sugar, like galactose. This is good stuff. You're going to love this if you don't already. All right. Any questions about that? Okay, cool. 4th question. How are prokaryotes unique? How are prokaryotes unique? What do you know right off the bat they're going to be missing? They're going to be missing all organelles, and especially the nucleus, because the name sort of tells you first nucleus. You're like, I don't really know what that means, but I think there's going to be something about the nucleus. All right. So the middle of this cell, they still have genetic material, because every cell contains what? Nucleic acids, right? Every cell, every living organism has DNA. And so they still have DNA, but it lacks a membrane surrounding that DNA. The DNA is just exposed. Everything in the cell can access the DNA. So that's unique. Most of these prokaryotes have a single circular chromosome. How many chromosomes do our cells have? 23 sets of 2 for a total of 46. Okay? So our cells have 46 chromosomes. A bacterial cell typically has a single chromosome, and it's circular. What are our chromosomes? Are they circular? No, they're X-shaped when we actually see them with light microscopy, but they're linear. They look like rods. On top of this, many prokaryotic cells have a cell wall. Cell walls are not unique to prokaryotic cells because what eukaryote has cell walls? Plants, right? And also fungi. But a cell wall made of a glycocalyx. And so glycocalyx, what does this root glyco mean? Sugar, right? So we had like glycoproteins, and those were sugars mixed with protein. So what do you think about the cell wall? If it's made of sugar, what does that mean? It is tasty. I mean, that came from here. I mean, it would be tasty. It is going to be polar. But what are sugars? Like, you're going to make a gingerbread house. What do you use to glue it all together? Use a nice sugar-rich icing, right? Sugars are sticky, right? You know what I'm talking about? Sugars are really sticky. If you don't know that, take a piece of fruit, smash it all over your face, and then feel your face. It's sticky, right? You're like, why do I have to put it on my face? Can't I put it on my hands? Whatever, okay? But it's sticky. This is weird. You're like, why on earth would a prokaryotic cell have a sticky wall? So it can stick to other things. Oh, this is good. Because bacteria, wherever bacteria live, they need to stick to other bacterial cells and stick to whatever they're attached to. Or if the cells, let's say, I don't know, let's just say hypothetically that you have nearly as many bacterial cells in your body as you do human cells. That's not hypothetical, by the way. That's actually true. You have nearly as many bacterial cells in your body as actual human cells, which really forces you to ask the question of, like, can you be human without those bacterial cells? It's another question for another time. But almost all of those bacteria are lining your intestinal tract. And they're nice and sticky, as some of them. Some of them have a membrane outside of that sticky wall. And so it helps them stick in place. So that when you've got this bulldozer that is high fiber, right? We talked about that before. That bulldozer coursing through your intestinal tract, or the garden hose, right? If you consume lactose when you're lactose intolerant, we're bringing all these images back. The bacteria stay put. They're not bulldozed out. This is just wonderful. Oh yeah, and so what this forms is like a slime layer or a capsule around the bacteria. Many species of these move their entire body, because their entire body is a single cell, using a flagellum. Only very, very small cells can move using a flagellum. Only very small cells. So it's unusual. It's unusual. You find very few, and what I mean by few is not few number of species, but very few types of eukaryotes that move using a flagellum. And then one last thing that is unique to prokaryotic cells are these structures called pili that actually function to anchor two cells together. And some of them are hollow inside and can be used to move genetic information, and so we call them sex pili, because they're transferring genetic material from one individual to another. And so bacteria transfer genetic information like crazy, which is one of the reasons why bacterial or antibiotic resistance develops so quickly. Because if you've got one individual that's antibiotic resistant, it can very easily transfer that information to another individual. And now you've got two individuals that are antibiotic resistant. All right? Any questions about this? I'm going to show you a couple of pictures of prokaryotes, and then we'll be done for the day. It is already almost 12:40. I know, it's just, man, Dr. Elaf flew by. I get it. Actually, I only have one picture to show you. And so here is the outside of a bacterial cell. Here you can see this genetic material called the nucleoid just hanging out, not bound by any membrane. Nothing's going to hold it back, right? And then here you've got inside a cartoon version of here. Here's the nucleoid, that genetic information. Here's a flagellum. Here are these pili that are going to function to do what? Anchor it to other cells or maybe transfer genetic information. All right? Have a wonderful conclusion to your Wednesday. Be safe. Make wonderful decisions.